Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to analyze ChIP-seq data. So what is ChIP-seq? ChIP-seq is a technique that allows us to study the interaction between proteins and DNA. This technique is crucial for understanding gene regulation, epigenetic mechanisms, and chromatin structures. How does it work? First, we cross-link proteins and DNA in the cells. That means we fix the interaction between proteins and DNA. Next, we lyse the cells and fragment the DNA-protein complex. In the next step, we introduce antibodies that bind to the proteins of interest. This is the core step of ChIP-seq. The quality of antibodies can directly determine the quality of ChIP-seq. These antibodies are associated with magnetic beads. These beads can get pulled down by strong magnets, and this is where the name ChIP comes from, chromatin immunoprecipitation. Once the complex gets pulled down, we wash off the proteins to harvest the purified DNAs, and these DNAs are then subjected to sequencing. The data analysis process for ChIP-seq is not much different from the ones for RNA-seq. Basically, it's adapt trimming, aligning, and quantification, except we call the enriched region peaks instead of genes, signals instead of counts. To analyze ChIP-seq data, we require a powerful Linux environment. Here, I suggest you use the HPC system of your institution, especially when you have multiple samples. If you don't have any access to HPC, don't worry, I am building one for you. So just stay tuned. Our first step is to set up the Conda environment and install all the essential tools. So just open your terminal, create a new environment called env HOMER, activate the new environment, set up all the channels, and install the tools using Conda install. -y means yes to all. No need to take notes. Everything is on my website, ngs101.com. Once everything is installed, we can start installing the most important pipeline for ChIP-seq analysis, HOMER. HOMER is so comprehensive that it can basically do anything for ChIP-seq analysis, from peak calling to peak quantification, annotation, motif analysis, goal enrichment analysis, visualization, you name it. So just create a new folder for HOMER, download the configuration script using Deduplicate, and install the script using Perl, and add the newly installed HOMER to the path of our new environment. And here we need to install the reference genome too, but HOMER has all the genomes built in, so just need to list all the genomes that HOMER has using Perl list and select the reference genome that suits your study. Here we use the human genome HG38. Once HOMER is installed, let's restart the HOMER environment to activate everything. The aligner we use here is called BWA. Like all other aligners, BWA requires a reference genome index. Fortunately, RefGenie provides a pre-built BWA index, so we just need to download the files. So create a new folder for BWA index and download the files using wget command. Here, we just store the URLs into two variables, so we don't have to copy this long URL every time we download a file. Our example data are from NCBI GEO database. Hopefully, you still remember how to download raw data from GEO. We first fetch the SRA file using prefetch, and convert the file into FASTQ file using FASTQ dump. Here we download two samples, one experimental sample and one control sample. We compress the FASTQ files and remove the SRA files to save the space. And then we rename the FASTQ files for demonstration purpose. Once everything's set, The workflow is much easier to follow. You just need to pay attention to the input and output of each step. Our first step is to trim the adapters using our old friend trim_glor. The input is a FASTQ file. Note the ChIP-seq is single-end sequencing, so there is only one FASTQ file. The output is the directory we created above. So we do the same for the control and the chip sample. Next, we align the reads to the reference genome using BWA. Note we set the BWA parameters in the very beginning. This is the path of the BWA index on your computer. This is the number of CPUs we're going to use in BWA. This is the format of calling BWA. The input is our trimmed FASTQ file. The output is an alignment file called SAM file. -T means how many CPUs we're going to use. -M labels the secondary mapping. -R, you don't need it. It's just tag. It shows the origin of the reads. And the reference is this parameter. It's the path of the BWA index. And we do the same for both samples. Next, we convert SAM files to BAM files, because BAM files are the standard inputs for many bioinformatics tools. We use samtools view for this task. Keep everything same as I showed you here. Focus only on input and output. The input is our SAM file. The output is a BAM file. Just give it a meaningful name. We do it for both samples here. Next, we sort the BAM files using samtools sort. The input is our BAM file. The output is a sorted BAM file. Just give it a meaningful, consistent name. If the memory is too small, just change it to bigger memory, say 16 gigabytes or 32, something like that. The next one is to index the sorted BAM files using samtools index. The input is a BAM file. You don't have to specify the output because the output by file will be saved in the same folder with BAM files. This step is new for ChIP-seq. It's quality control after alignments. Basically, we want to remove PCR duplicates and filter out blacklisted regions. We remove duplicates using Picard mark duplicates. The input is our sorted BAM files. The output is our deduplicate BAM file. Just give it a consistent name. The M option is optional. It's for debugging purpose, so you don't need it. Remove duplicates. True, yes, we do want remove duplicates. Validation stringency, we want it to be lenient. You don't want to remove too many things from your BAM files. We do deduplication for both samples. And don't forget to re-index the new BAM files. Next, we filter out the blacklist regions. So what are blacklisted regions? They are unstructured DNA sequences in our genome. For example, repeats. So we don't know the meaning of the repeats. So if reads map to these regions, we can't explain. So we download the blacklist of human genome from this URL and overlap our BAM file and these blacklist regions. We only keep the regions that don't overlap with the blacklist. And then when we save the results into a new BAM file, it's called dedup filtered BAM. Just give it a consistent name. We use better tools intersect. -V, you need this option because we only need the non-overlapped region. ABAM is input, our BAM file. -B is input, the blacklist region we just downloaded. Output is a filtered BAM. Just give a proper name. We do it for both samples and re-index the new BAM file. Finally, we're ready to call the peaks using HOMER. Before that, make sure you have created all the necessary folders for the results. First, we call the function make tag directory for both samples. The input is our QC BAM files from last step. The output is the tag directory we just specified over here. We have specified the genome is human genome HG38. After that, we can call the peaks using findPeaks. Two inputs, one, the ChIP sample tag directory we just created, and the control tag directory from the last step. The output will be the peak file. Give it a consistent name. Style factor, because we're doing transcription factor ChIP-seq. FDR set you 10 to the minus 3 or 10 to the minus 5, depends on how stringent you want it. After that, we can annotate the peaks using annotatePeak.pl function. The input is the peak file from the last step. Human genome, specify it, HG38, you don't have to do goal enrichment analysis. The output is the annotated peaks in this format. The coordinate, the peak ID, the annotation, the closest gene name, and all the distance to the TSS and all other annotation. So with this, you can do a lot of things downstream analysis. And that's about it. Subscribe if you like it, and I will see you in the next video.